In this video we're going to take a look at some of the ways in which you can use the tools available in SeqMonk to analyse large single-cell RNA-Seq datasets. These types of study are becoming more popular and they have some challenges which are pretty much unique to that type of data. And here we're not intending to give you a complete guide to how to analyse single-cell RNA-Seq, but simply point you at some of the ways to apply the existing tools that SeqMonk has in ways that have proved useful on these types of dataset. The dataset I'm working with here is quite a big single-cell dataset. There's around 400 individual cells loaded in here. So if I look at my datasets, you'll see there are a lot of them in there. At the moment I've only got a few of them showing in the chromosome view because obviously there's not enough space to put them all in, but you can just get an idea that this looks pretty much like normal RNA-Seq data. So the import for this is just standard RNA-Seq input, the same way you would for normal bulk data. There's nothing special about that. The one unusual component of this dataset is that in addition to the normal chromosomes, this was a dataset which had ERCC spike-in controls. So this has an extra ERCC chromosome. I'll show in a separate video how you actually set up these kind of multi-genome projects so that you can incorporate ERCC spikes, but once the data is imported it just appears as another chromosome and the ERCC spikes are in there. At the moment we have the raw data straight off the sequencer in here, and before we start doing any analysis we want to do some QC on this. The QC on the data in single-cell RNA-Seq experiments can be very variable, and in particular what you often find is that there will be a subset of cells whose data is very poor and which you want to remove from your analysis at a very early stage, otherwise they'll start adversely affecting the analyses you do downstream. The easiest way to get into this is to simply go into the plots menu and do the normal RNA-Seq QC plot, the same as we would for any other RNA-Seq data. I'm going to select all of my cells and just run with the default options. I'll pause the video at this point because it'll take a little while for this to compute, so you don't need to watch that, but I'll bring it back again when it's finished. Okay, the plot has now finished calculating, and if I expand this out a bit, there's obviously way too many samples here for you to actually be able to do anything sensible with the names down here, but that's not what's important in this plot. What we have are various metrics that we've calculated from the data, so each one of these panels is a separate metric, and what we should be expecting to see is that for the most part all of our samples look very similar to each other. And what you'll see is that in the case of these single cells, whilst a lot of the cells will have similar values to each other, there's an outgroup in most of these plots, or maybe two outgroups, where you have aberrantly behaving cells. What you'll also find is it tends to be the same cells which are aberrant in all of these different measures. And what we want to do is to tag and remove those poorly behaving cells because they're probably going to have some kind of technical artifact. So if we look at these, things that we're seeing is that we have cells which have unusually low percentages of their data falling inside genes, which is isn't what we'd expect for RNA-Seq, that have either unusually high or unusually low proportions of genic data falling into exons. The ribosomal RNA contamination is not too bad, but we've got a few samples that seem to suffer from that. Percentage of genes measured is quite a key value to look at, because again you want this to be quite similar for your samples, and here we've got an outgroup which have very small numbers of genes measured. The percentage of max data size is much more variable generally in RNA-Seq, but again we've got a set at the bottom which just have very small amounts of data. The ERCC spikes, these can be used for sort of quantitative normalization, but actually what we've found is that the easiest use for them is to simply flag up poor quality cells. So where the ERCC spikes make up a very high proportion of all the reads that are in your data, that's a big red flag for the data being of poor quality. Mitochondrial sequences, again we've got a few, and a little bit of strand bias in some of these. Now the way we can use this plot is that having plotted the data out this way, we can now drag the mouse inside any of these plots to select sets of cells. So if I drag in here, I can select all of the cells which are behaving poorly in this, and you'll see the corresponding cells highlighted in the other plots as well. So you can see that the outliers here are also outliers in a lot of the other plots, and then I can tag these. So tagging will simply put a prefix on the names of these samples. So the default is bad QC with an underscore, so I'll do that. I can do the same thing in the next plot, so I can highlight these, get rid of those, do the same thing in here, get rid of these. And just work my way along. So I'm going to go through all of these plots, tagging poorly behaving cells, Okay, so I've now gone through all of these panels and have tagged the bad quality cells in here. Having done the tagging, I then want to get rid of those out of the project. So if I close this down, I can now go into my datasets here, and I'll see the samples that have got bad QC tagged onto the front of them. So there's a set of those, but then most of the data is still okay. Okay, so what I'm going to do is to select the bad quality cells and just delete those out of the project. Okay, so I've taken 99 datasets out of here. Okay, having done some basic QC, I now want to go on and quantitate the data, so that's what I'm going to do next. The quantitation I'm going to do here is a completely standard RNA-Seq quantitation, so just in the data menu, go to quantitation pipelines, select the RNA-Seq quantitation, keeping all the defaults. These are unstranded libraries, so there's no strand specific bias to take into account, so I'm just going to quantitate everything. Again, this will take a little time, so I'm going to pause the video while this completes. Okay, the quantitation is now completed. At this point I'm going to add all of the samples back into the view. So if I just put all of those in here, now, because there are so many samples, you really just can't see anything in here, but if you select a sample from the data view here, then the main view will actually expand that sample so that you can see what's going on in there. So even though there's not enough pixels on your screen to see all of them sensibly, you can at least see an individual sample okay. The next thing we want to look at is the distribution of the quantitation, so same as we would for a normal RNA-Seq. So in the plots menu I can just do a cumulative distribution plot. And what this will show me is that there are going to be a huge number of genes which are essentially unmeasured in all of my samples here. Okay, so you can see this sort of flat line across here are things that are unmeasured, and the only reason they don't sit at the same level on here is because the total amount of data in each one is different, so the limit of detection is different. I want to focus really on genes which are expressed at a reasonable level somewhere. So I'm going to just do a simple values filter. In this case I'll filter on a value of 3, so it puts us somewhere into the main part of the distribution in here, but I only care if that gene reaches that level of expression in any of my 300 odd cells. Okay, so I'm just going to do a simple filter just to remove the completely unexpressed stuff. So I'm going to filter across all samples, say the value's got to be above 3, but only in one of my 285 cells. Okay. And having done that, I'll select that subset. So we've lost about a third of our genes in that way. Now, there are a few different ways to look at this data, but the way that we're going to start off looking at it is to look at the variability between the different samples. In this data set, I don't have predefined groups of cells. It's all just one set of cells, and I want to see if there's any structure that I can spot from the data. And an easy way to get to that is, rather than looking at all of the genes, is to look at genes which are unusually variable, and therefore where the variability might reflect a difference in expression rather than just natural variability in a homogeneous population. In order to do that, I'm going to need to make a replicate set, and a replicate set which has all of my cells in it. So I'm going to do that first. So I'm going to go to Edit Replicate Sets, make a replicate set just called All, and then add all of my cells to that set. Now I'm going to use the variation plot that we have in SeqMonk to look at the variation of the expression of each gene across all of those cells. The variation plot allows us to look at variation in a couple of ways, and we'll plot a couple of these out, but probably the easiest one to start with is just to plot out the standard deviation. Okay, so what we can see in here is the expression level of our genes on the x-axis, and then the standard deviation on the y-axis. And you can see that overall there is a relationship with this. The reason that this decreases at the start is because as your expression gets low enough, it hits the limit of detection, so you just end up with 0, which is quite a stable value. But thereafter, once you've kind of peaked through that, then your variance will also decrease as you get to higher expression levels, just because you're better observed and have less technical noise in them. The points that we care about are the points which show unusual variability. So I'm not sure how clear it is on the video, but you can see that on top of this cloud of points there is a regression line that's being calculated, this black line that goes through here. So by looking at the distance away we are away from that black line, we can pick out points which have unusually high variance given their expression level. Okay, so these points up here that sit out from the cloud are the ones that we really care about. We can actually select these directly. So if I look at this, if I put my mouse on a point up near the edge of the cloud, at the bottom down here it shows me the difference from the regressed mean. So I'm getting a difference of about 0.7 of a standard deviation away from this center line to give me these sort of outliers that I'm interested in. So I'm going to go and select those specifically. Okay, so if I go into the filtering menu and say filter on variance and individual replicate sets, in this case I'm going to filter for things which have a relative standard deviation more than 0.7 of a standard deviation away from that regressed line. So I'm going to correct against local variation, so it's the difference from the regressed mean on here. Okay, she'll calculate that, so I get just over 300 genes coming out, and I can then highlight that on here just to check that I'm seeing what I expect to see. There you go, so those are the highly variable points that I think are likely to be informative in separating these cells out. While we have this plot open, we can also show another way of looking at this data. So this is plotting out the standard deviation. Another way that's interesting to look at single cell data is to plot out this unmeasured value. So what we plot on here again is, on the x-axis is still the expression level, and on the y-axis we plot the number of cells for which we didn't have any data. Okay. So in general we expect that there is a relationship with this. If you have lower expression, you should have a higher proportion of your cells being unmeasured because you're at or below the limit of detection quite frequently. But again, the points that we're interested in are the ones where we see individual genes having an unusually high proportion of their cells showing that gene as completely unmeasured, whereas when they are measured they have a reasonably high level of expression. So in this case again we want to pick the cells off the top. We can have a look and see if the variable genes that we picked before are also relevant in this plot. So if I highlight those on, again you can see that they do fall on the top of this view as well. So we could have made the selection directly on this data, so in the same way when we did our variance plot we can filter on this data, but there should be a very high degree of correlation between this information and what we saw from just plotting the standard deviation. In this final part of the analysis, we're going to take this set of genes that we identified as being unusually variable and then use some of the clustering tools in SeqMonk to either see if the genes themselves can be subdivided into groups which have similar behavior, or whether we can subdivide the cells based on the expression profiles of these genes across the cells. So we're going to look at both of those ways of doing clustering. We'll start with clustering the cells. So to take a simple look at the clustering of cells, we can go into data store similarity and draw a data store tree. So this will show us the overall correlation of the values for those interesting genes across all of our cells. And we can see here that this tree has some significant structure to it. So there are two very large groups of genes here, where we can see the separation of there, but then there's other sort of subgroups within that. So we're going to do two things from here. So firstly, we're just going to reorder all of our data stores so that they match the structure in this tree, so that when we do some plots later on, the cells will all be in the right order. And then secondly, I'm going to partition this tree, so I'm just going to put a split point in here and then split the data stores, and that will make me some sets of cells that all behave similarly across this set of genes, and we'll see what we can do with those later. Right, the next question then comes as to whether there are subgroups within this cell, within the genes that we've identified here, and to get at that we will do a hierarchical clustering. So if I go into plots and hierarchical clusters and per probe normalize, so this will do me a correlation based clustering of these genes across all of my cells. It'll take a second to calculate and then we should be able to see. Okay, so if I just turn these splits off and adjust the contrast a bit, what you can see in here is that there is some very obvious structure to the genes themselves. So the genes are running down the y-axis here, and you can see that there are, well, at least three groups. So there's a big group up here, which are kind of high in this set of cells, low in this set. There's an opposing group which is low in this set of cells, high in this set, and then there's another group down here which does a patterning which is not really related to those other two groups. If I bring up my divisions here, I can find an appropriate split point to try and separate those three groups out. So here I can see that I'm separating 1, 2, 3 groups of genes. So if I save my clusters, I've actually got a fourth one that's come in. There must be another smaller one in there, but I now have the two major groups of genes which I saw, the third smaller cluster, and there must be a fourth very small cluster that came through from one of those sets. This immediately gives me something that I can start relating to biology, because each of these clusters now should have a similar expression profile across all of my samples. So for example, if I go and do a line graph across this, I can see that there is a defined profile for all of these genes across my set of cells, so I can start investigating what biological functions might link the set of genes that are present inside this cluster. Okay, so I have a way directly to get into biology. So we now have a separation of both the genes in our interestingly variable set, but we've also split our cells by creating these automatically created replicate sets. So we have the 4 groups of cells based on the correlation tree that we made from our variable genes. So as the final way to look at this data, we're going to use a different tool. So we're going to go back to all of the expressed genes, and now we're going to plot out a PCA plot. Okay. So we're going to put all of these through a PCA just to see how they separate and whether there are multiple levels of separation that might be achievable on the cells. Takes a second to calculate, but then should come up. Okay, so within the plot itself we see a couple of things. So if we look at the principal components, we can see that the variance here is not very highly explained by any individual principal component, so there may be multiple effects going on inside here. We can also see that the first, second and probably third principal components are all reasonably interesting, and then it falls off somewhat after that. we've shown in a different video where we look specifically at PCA how we can actually use the rotation values associated with principal components to start separating genes out, and that would be another way to get at a set of interesting genes, but we're not going to follow that up for the moment. What we are going to focus on is the actual PCA plot. So within here we can see that again there are suggestions of multiple groups of cells in here. Okay, so maybe one over here, and one down here. So what I'm going to do is to highlight on the groups that we defined from the correlation tree and see if that matches up with what we see in the PCA plot. So if I look at that, what I can see is that there are some very clear groups in here. So the groups that we defined from the correlation tree do seem to do a reasonable job of separating out in the same way that the PCA plot does. So we have the red group over here, which is group 2, the blue group over here, which is group 1, then two groups down here which are not particularly well separated on the PC1 or PC2 that we look at here. So the green and the orange, they do separate slightly but not brilliantly. So if we look at a second one, so if we look at PC3 we see actually separates the green and the orange really quite effectively, so that's the main division of these two. And we can even look down at further principal components. PC4 separates off the blues from the remainders. So these two approaches are both ways of being able to look and validate any subsetting of cells that we might pick out. And now once we've got these sets we can start investigating these individually, and if we want to dig into this analysis some more, we now have the groups of cells that we can start looking at to see how they behave and what might be different about those, and to characterize the groups of genes against groups of cells. Okay. That's a bit beyond the scope of this particular video, but hopefully this has given you a feel for how you can actually start getting into these data sets and understanding what can be quite complex biological questions.